The kidneys are able to filter large volumes of blood plasma by a process called ultrafiltration. This entails the existence of pressure gradients across the glomerular filtration barrier, facilitating the movement of small molecules from the blood plasma within the glomerulus and excluding the movement of larger molecules such as proteins and blood cells. The purpose of this video is to discuss the main determinants of this filtration process and to also describe how these individual parameters can determine the glomerular filtration rate. The glomerular filtration rate, or the GFR, describes the rate at which blood plasma is being filtered from the glomerular capillaries into Bowman's space. The two main determinants of this glomerular filtration rate are the net filtration pressure, or P-net, usually expressed in millimeters of mercury, and the capillary filtration coefficient, milliliters per minute per millimeters of mercury. This filtration coefficient is proportional to the surface area of the filtering membrane, the glomerulus in this case, and its fluid conductivity. It's worth noting that the glomerulus is an extremely conductive capillary bed. The filtration coefficient for a typical glomerular capillary is approximately 400 times larger than a normal systemic capillary bed. So the glomerular capillaries are specialized for the formation of ultrafiltrate. Although there are instances where the filtration coefficient can change either during normal or pathological conditions, the main regulatable parameter that influences the glomerular filtration rate is the net filtration pressure. The net filtration pressure is determined by the balance of Starling's forces. The two main components that determine the net filtration pressure is the hydrostatic pressure, which is the pressure exerted by a fluid in a confined space, and the oncotic pressure, the osmotic pressure exerted by dissolved proteins and associated counterions. Because we're talking about the movement of fluid from two separate chambers, that is, between the lumen of the glomerular capillaries and Bowman's space, these two pressures can be divided into four main components. First of all, we have the hydrostatic pressure within the glomerular capillaries. This essentially is the blood pressure within the glomerular capillaries. And also there's a hydrostatic pressure within Bowman's space. This hydrostatic pressure is essentially a back pressure as a byproduct of the tubular fluid moving through the nephron. There's also the plasma oncotic pressure within the glomerular capillaries, largely due to the presence of dissolved plasma proteins such as albumin. And then the potential for an oncotic pressure within Bowman's space. This is usually negligible due to the low concentration of dissolved protein within the glomerular filtrate, but it's worth noting that this parameter can be increased by certain pathological conditions such as glomerular nephritis. In order to see how these individual pressure components can determine the net filtration pressure and then ultimately the glomerular filtration rate, we need to organize them between those pressure components which tend to favor filtration and those which tend to oppose filtration. The pressure components which favor filtration are those which tend to promote the movement of water and other dissolved solutes from the glomerular capillaries into Bowman's space. These pressure components are the glomerular hydrostatic pressure and the oncotic pressure within Bowman's space. Again, because that oncotic pressure within Bowman's space is usually negligible, it is the glomerular hydrostatic pressure which tends to be the main driving force for the movement of fluid from the capillary beds into Bowman's space. Conversely, the pressure components which tend to oppose filtration are the glomerular oncotic pressure, which exerts an osmotic pressure that tends to pull fluid from Bowman's space into the glomerular capillaries, and then also the hydrostatic pressure within Bowman's space. That back pressure exerts a hydrostatic pressure that tends to push fluid from Bowman's space into the glomerular capillaries. The overall net filtration pressure is determined by the magnitudes of each of these individual pressure components. So different ways of organizing these components to determine what net filtration pressure is. I like to use this method where I take the sum of the pressures which tend to favor filtration and subtract from them the sum of the pressures which tend to oppose filtration. Let's walk through an example using some reasonable pressure values. For the forces which favor filtration, a reasonable glomerular hydrostatic pressure is approximately 60 millimeters of mercury. And as mentioned before, the oncotic pressure within Bowman's space is typically negligible, so approaching 0 millimeters of mercury. And for the forces which oppose filtration, you could expect the hydrostatic pressure within Bowman's space to be approximately 18 millimeters of mercury, and the oncotic pressure within the glomerulus to be approximately 32 millimeters of mercury. If we sum each of these groups of components, we get 60 millimeters of mercury minus 50 millimeters of mercury, or a net filtration pressure of 10 millimeters of mercury. So this net filtration pressure of 10 millimeters of mercury determines the driving force for fluid movement from the glomerular capillaries into Bowman's space. It might not seem like much of a driving force of only 10 millimeters of mercury. When you consider the capillary filtration coefficient of the glomerular capillaries, this 10 millimeter of mercury net filtration pressure is actually quite significant. A reasonable filtration coefficient for 2 kidneys in a 70 kilogram animal is somewhere in the neighborhood of 12.6 milliliters per minute per millimeters of mercury. The glomerular filtration rate is simply a product of these two parameters, the net filtration pressure and the filtration coefficient, which in this case produces a glomerular filtration rate of 126 milliliters per minute. This is 126 milliliters of blood plasma moving from the glomerular capillaries into Bowman's space every minute. This amounts to the filtration of a little over 1 liter of blood plasma for every 8 minutes. This is a tremendous amount of fluid that's being filtered by the kidneys. Although this falls in the normal range of a GFR for a typical 70 kilogram animal, it's important to note that the GFR itself is a regulatable parameter. There are a number of factors and regulatory processes which can cause the GFR to increase, and other regulatory factors and processes which can cause it to decrease. These processes play an important role in the kidney's ability to produce appropriate compensatory responses to various body fluid challenges. These processes tend to alter the glomerular filtration rate by modulating the net filtration pressure. So again, there are 4 main pressure components which ultimately determine the net filtration pressure. But the 2 that appear to be most readily regulatable by the kidneys are the glomerular hydrostatic pressure, which tends to favor filtration, and the glomerular oncotic pressure, which tends to oppose filtration. It should be readily apparent that factors which tend to increase the glomerular hydrostatic pressure will tend to increase the net filtration pressure and therefore increase the glomerular filtration rate. Conversely, factors which tend to decrease the glomerular hydrostatic pressure will tend to decrease the net filtration pressure, thereby reducing the glomerular filtration rate. These changes in net filtration pressure can also be accomplished by modulating the glomerular oncotic pressure, where either increases or decreases in the oncotic pressure can result in their attendant changes on the glomerular filtration rate. So how is it that the kidneys can exert changes on the glomerular hydrostatic pressure and the glomerular oncotic pressure? Please check out my subsequent video where I examine these relationships and discuss how changes in the afferent and efferent arterial resistances can exert control over the glomerular hydrostatic pressure and the glomerular oncotic pressure.